Okay, this lecture is recorded, and so you are able to access it on the YouTube channel. A couple of things I do want you guys to make sure that you do this semester is that you look at the syllabus. Make sure you review it, because some things you thought are not true. And you'll find that out if you review the syllabus. Now, it's called Course Policies on the AMP website, correct? It's not called syllabus, it's called Course Policies. So please make sure you review the 228 course policies. Something else that you have to realize, you have 227 and 228 group access, correct? Have you guys realized that? Because I'm seeing a lot of posts from people saying, I can't see 228. I'm just going, I look it up and it's like, you got 228 access. You have 227 and 228 access. And you guys noted that in under resources, when you take without clicking and making something appear, you have the big window that opens up, right? And it says lecture resources and lab resources, and it lists it twice, doesn't it? The first lecture and lab resource is for 227, and the second lecture and lab resource is for 228. And you guys should also see off at the right-hand side forum postings for 227 and 228. And if you see a new 227 student posting and you would like to reply, kudos for you, because I greatly ask you to do that, because they listen to you guys more than they listen to me, even though they should be listening to me. So, and I always appreciate help, because one of the things that I really do think is we are all a team. You guys have survived first semester. You now here in second semester, you know how I work, and you know how I want you to be successful. And so success is always a challenge, and that's just kind of part of any sort of learning process. In lab, you guys will be going down to the computer lab at the beginning. You meet up in the lab, the regular lab in the beginning, then go on down to the computer lab to make sure that you get your Connect access established. How many people have already done that so far? So, yes. And getting that Connect access established will also give you your what I call your carrot. You get a point for getting that established during that lab time. So make sure that you know your password, because some of you are, you know how we do with everything at home? We have the computer and it remembers everything, and so therefore we forget everything else. That's right. So you guys have to remember, don't let the computers take over your life. They're supposed to augment your life, not to take it over and become your brain, because without the computer you'll end up actually stumbling quite a bit. But that's a lesson that I have to experience all the time, because all my passwords are on my iPad here. So, you know. So I realize I need to write them down someplace, because my iPad one day will die on me. Heaven forbid. But you know how technology is. All technology does die on you when you least expect it. Right? Haven't you had something go fritz on you when you least expected it? So keep that in mind too. Your technology does that to you. Your lab is over special senses, and that's what our first lecture, several lectures are over, is actual special senses. Now, previously we spent how many chapters? 4 chapters on the nervous system. So this is our 5th and final chapter on the nervous system. Special senses differ from our other sensory receptors, because these are complex sensory receptors. We had a distinction. We had simple sensory receptors and complex sensory receptors. And it's because the simple sensory receptors are simple in their construction, and the complex sensory receptors are complex in their construction as well as the information relayed. Because if you're simple in construction, the information must be simple. If you're complex in construction, the information relayed is actually much more complex and has a wide variety of actual perceptions. Because isn't that what everything is? Everything's all about perception. So simple construction, complex construction, simple sensory information, complex sensory information. So that's what we're dealing with. And the complex sensory information comes from our special senses. And we have smell, right, which is olfaction. We have taste, which is gustation. We have vision, which is what nerve controls that one? The optic nerve. And then we have balance and hearing, and that's the vestibulocochlear nerve, cranial nerve 8. So we will go through all of those special sensory receptors because something has to happen to them to get that information transmitted to the brain. We're starting off with olfaction, which is sense of smell, and gustation, which is taste. Let's talk about the olfactory neurons. They're located in the olfactory epithelium, and that is located in the roof of the nasal cavity. Now, the nasal cavity's opening is the nasal aperture, or also commonly called your nostril, and that's an anterior nasal aperture. An aperture is an opening, and we also have posterior nasal apertures, and that's why we actually have some of that distinction. You might run across that anterior nasal aperture, posterior nasal aperture. And what typically happens when we breathe through our nose, because we are actually born as obligate nose breathers, you guys may have noticed a newborn baby always breathes through its nose, and that's why when they have a head cold, it's so distressing, because they actually experience respiratory distress, correct, when they actually have a head cold, because they are obligate nose breathers. That's the wiring pattern that is set up for when you were born, because you're going to breathe through your nose as you feed, as you breastfeed, because that's the pattern of eating for them. And so when they get sick, they have a lot of difficulty figuring out how to survive when they have so much nasal congestion and nasal mucus in them. So one of the things that's important is our olfactory epithelium is between our eyes, and our nasal aperture opening, our anterior one, is down here by our mouth. So when you breathe in, oh yes, sorry about that. You breathe in, hang on. I love having fun at the computer. When you breathe in, the air flows like this. Goes in the anterior nasal aperture and out the posterior nasal aperture. And so when you actually are trying to smell, you don't get a lot of molecules that are containing of the chemical that you actually want to smell or appreciate coming close to that area. And so it's an interesting thing. When you look at somebody's nose, they have things coming into the nasal cavities from the side. These things that are coming in from the side are called nasal concha or turbinates. And they are designed to help vortex the air that's moving in. But still, the air wants to go over the hard palate and the soft palate and out the posterior nasal aperture into the area called the nasal pharynx. We'll get into more details about that later on. In order to get the molecules, the chemicals, up to the olfactory epithelium, we need to do a process that will actually increase the vortexing. And we do this all the time whenever we are trying to actually smell something. What do? We sniff. And so sniffing is our method to actually increase the vortexing so that we can get these molecules up to that olfactory epithelium. So our notes say epithelium is in the roof of the nasal cavity, poor location for smelling, and sniffing improves the ability to smell. Now, one of the things that's also interesting is you thought the olfactory bulb was where the olfactory neurons, or cranial nerve 1, was actually located. It's not. It's in the olfactory epithelium. So take a look here. It says olfactory receptor cell, often called neuron, is the actual location. And these guys are in the olfactory epithelium surrounded by support cells and basal cells. And they have their receptive end obviously in the nasal cavity area, correct? And then they have in the area where the basement membrane is, that's the area where they're actually on the undersurface of the cribriform plate, and they send their axons through those little holes in the cribriform plate up to our olfactory bulb. You remember the olfactory bulb in the sheep? Well, the olfactory bulb in the human is so much smaller. So what does that tell you about the ability to smell different things and the sensitivity to different things? Who has it better, human or the sheep? Sheep. And also, what do you think about dogs? Dogs are better than human also, because the bigger real estate you are given for any one thing, that means the more information that you are actually going to send to your brain for that. So ours is pretty small, but we don't depend upon our sense of smell for survival, do we? For survival, we depend upon vision for this. And so that's why when we get into the eye, that's a very important structure for us, because that's where our survival actually is dependent upon. But sense of smell, that's not necessarily all that important for us. It kind of augments our experience of life, but it's not necessary to live a good life and to survive. When you take a look at this cartoon of our olfactory epithelium, we have in the nasal cavity, we have a mucus layer, and then we actually have hairs coming off of our olfactory neurons. And we have this area called the dendrite. Then we have an area called the cell body, and then we have an area called the axon, which goes through the foramina of the cribriform plate. Now, dendrite means what to you guys? Receptive end. So that makes sense, right? Isn't that where the chemical needs to come into contact? So realize they named this based upon where things start. So we start at the dendritic end, then the information goes to the cell body, and then it goes to the axon, which will then synapse in the olfactory bulb, and the information will go to various areas of our brain for that effect. So keep that in mind, that we have this inverted look to this, but that's because it's inside the nose, and it's also, as I said, in a very odd place. Olfactory receptor cells, or notes say, are also called olfactory neurons. They are chemoreceptor cells that are actually unusual in that they are bipolar neurons with these things called olfactory hairs that are non-motile, so they're not really hairs. We call them hairs because of their appearance under the microscope. What's interesting about these bipolar neurons is these guys are replaced when damaged. What do we know about neurons when you damage a neuron and kill it? It's gone, right? Well, our olfactory neurons are bipolar neurons, and when they are damaged, they are actually replaced, and they are replaced by basal cells. The basal cells undergo differentiation and replace the olfactory cell. So this is an unusual situation, because most neurons are not replaced. And you know that this has to occur because when you have a bad head cold, what happens to your sense of smell? It goes away. And it goes away because those olfactory neurons were actually damaged and die. And so it takes a while to recover your sense of smell because those basal cells have to differentiate then into those olfactory neurons to actually give you that information. Support cells, they form the bulk of the epithelium, and they do what it sounds. They support the epithelium. They also produce mucus. PD is my abbreviation for produce mucus. This is protective for the olfactory epithelium. It keeps everything moist. So the reason we need to keep things moist is, what happens to you if you sit by a fan all day long? You dry out. So what are these guys doing? Air is running by them all the time, so we need to be able to provide an extra barrier of moisture so that they actually don't dry out and actually then decay or die. Now, basal cells are also known as olfactory stem cells. They are located at the base of the epithelium, and they They are the cells that replace damaged olfactory neurons. It's absolutely fascinating. We can detect 1 trillion different odors, but we all are not constructed the same, and I'll tell you a story about that in just a moment. We can detect 1 trillion different odors, but we only have 400 smell genes. So what does that tell you? Those 400 smell genes, your odors, what you can detect or appreciate or acknowledge in your cerebral cortex, is based on the combination of activation of these smell genes. It's like painting, and I've only given you 3 colors, red, blue, and yellow. What do you know you can do with those 3 colors? You can create a variety of colors, correct? And that's what these smell genes do. That's why we have 400 smell genes, yet we can actually detect 1 trillion different odors, because these chemicals will activate various receptors, and the various receptors you activate give you then that appreciation for those all different smells or odors that you actually experience. So a smell gene encodes for a specific receptor protein on the olfactory hair, because that's where the receptors are located. The receptors are located on the hair. So it's not a real hair, but we call it a hair, and that's where you find receptor proteins. Now, think of our synapse, because somebody made a post that I had not explained it well enough for them to understand it during lecture, and so I'm going to work on my explanation with you guys a little bit better. Remember synapse. We release a chemical at one end, right? And the chemical diffuses across the synapse and lands on a receptor, which is a protein. It's a transmembrane protein at the synapse, and when the receptor binds to that, when the acetylcholine, for example, binds to that receptor, what happens? It opens the sodium gates, and sodium is actually allowed to actually then enter the cell, and if enough sodium enters the cell, then we get threshold, and then we get an action potential. And so for us, I'm going to make us come on down here. For us, we're in the sensory aspect of the nervous system, and we have to transduce. Transduce means transfer a mechanical event or a chemical event into an electrical event. Okay, so all of our sensory receptors have to undergo this phenomenon of transduction. So it's a little bit different from that synapse, but not enough to where you cannot relate to this receptor protein is responsible for the chemical to come in contact with, and if only one molecule lands on that receptor protein, will you get an action potential? If 2 molecules land, will you get an action potential? It has to be a certain amount, enough to land on our receptor proteins for us to get what in the world of the sensory receptor is called a generator potential, because generator potential, remember, is synonymous with threshold. So when you get enough chemical to land on those receptors, you always get a receptor potential, right? Because that means that a chemical landed on it. Okay, when you get a receptor potential, if enough chemicals land and give you enough receptor potentials, you will get a generator potential, which always means then you get an action potential, and that information then goes to the cerebral cortex. So let's see, back to the smell genes. Encodes for a specific receptor protein on the olfactory hair, and this is going to respond to a limited or select number of chemicals. And it is the combo of activation or activations that give the wide variety of perceptions. In other words, the possibility to experience 1 trillion different odors. How many of you like asparagus? Raise your hands. It doesn't hurt. Gives you something to do. Besides, right? Okay. Now, when you eat asparagus, if you eat more than 3, I think that 3 is the cutoff, and you go to the bathroom, your pee is not normal. Your pee is kind of green in its tinge, and many of you know this, but there's some in this room that do not know it has a distinct odor. The distinct odor is from a chemical called asparagine. Okay. And one of the things I hope that you caught on that I was hinting to is that some people cannot smell asparagine. So some people do not know that their urine smells when they eat asparagus. So what do you know about that person and the gene for that chemical receptor? They don't have it. That's right. So that's the other thing that you don't realize. We have 400 different genes, which give us a wide variety of odors, but not everybody has the same number of genes. They say 400, but we're not all the same. So you have to realize that some people cannot smell certain things, not because they have a head cold, because they have no idea what you're talking about because they were born without it. The same thing for the sense of taste. How many of you hate cilantro? I've heard that for you it tastes like soap.
Pine tree.
Pine tree, soap. Okay. For those of you that like cilantro, do you experience that? No. So that tells you that there's something about your receptors for the chemicals in cilantro. You have a different set of receptors and are missing certain receptors, and vice versa, correct? So if you don't have the receptor, that means you don't have the gene. Can you have the perception? So perceptions will vary based upon the receptors. And let me ask you this, if you have a lot of receptors versus very few receptors, who's the one that's more sensitive? The one with a lot of receptors. And so there will be those people out there that are super sensitive. Oh, I can't handle that. Well, you're being mean by saying, what's wrong with you? You know, what they're telling you is that they're very sensitive and you're not, aren't they? So you have to realize some people have more sensitivity because they have higher level of receptors and other people have less. Yes?
What happens in pregnancy that makes people smell stuff?
Yeah, and they still don't have good explanations for why pregnant women have certain food cravings and certain sensitivities to certain smells. They still, I keep reading about it, and nobody wants to do research on pregnant women and their sense of smell. Go figure. I don't know. Ladies, life isn't fair in the world of earning income equality as well as studies. So it is still a man's world, people, and yeah, everybody likes to complain, correct? That's all it is. It's just people want to complain. Okay, so activation of our olfactory cells, these things have to be true. The chemical must be volatile. What is volatile? Gaseous, in the gaseous state, right? So the chemical must be volatile, so that is gaseous. It must dissolve in water because the foundation of mucus is water in order to get to the olfactory hairs to get to the receptors. It must bind to the hairs because that's where the receptors are located. And once you get enough binding to the receptors, you will get that generator potential, which will always give you then an action potential. So that's the olfactory epithelium. Now let's talk about taste buds and taste, which is gustation. What's on the surface of your body? What kind of stuff is this stuff here on the surface of your body? Skin. And what is the, histologically, what do we call this stuff? Stratified squamous epithelium. It is cutaneous membrane, stratified squamous epithelium, keratinized, sitting on top of dermis, which is CT, right? It's CT, connective tissue. Okay, so now let's go inside the oral cavity. What's lining our oral cavity and even our esophagus? Stratified squamous epithelium, but it is not keratinized because the keratin prevents desiccation or drying out. And in the oral cavity, we have all kinds of spit and moisture and stuff like that. So we don't need keratin there. But it's still stratified squamous epithelium because we put things in our mouth and it's rough. Okay, so rough stuff deserve multiple cell layers for protection. So what's over our tongue, the surface of our tongue? Stratified squamous epithelium. So that's my point here. In the oral cavity, everything in the oral cavity, and all the way down the esophagus to the gastroesophageal junction is stratified squamous epithelium because we chew things up and they're rough and we are actually losing epithelium every time we eat, but that's okay. We just swallow it and, you know, if we can use it, we use it. And if we don't, we just digest it and poop it on out, you know, We've got a very cool system here. Yeah, we'll take it. Nope, out it goes. Yeah. So when you take a look at your tongue, it has, some people will describe it as a velvety appearance. There's all kinds of different looking tongues. You should actually get online and do a search of tongue appearances because there's some weird looking tongues out there. The geographic tongue, the black tongue, and all kinds of weird stuff. But overall, we'll just say it has a velvety appearance. The reason it has a velvety appearance, it is covered with, obviously, stratified squamous epithelium that is covering these structures called papilla. And so papilla are projections. You guys remember we use the word process for projections? Well, papilla are projections. Remember the dermal papilla? So that was that wavy area. So papilla are projections, and our papilla on our tongue, or what gives our tongue its velvety appearance, and they're all covered in stratified squamous epithelium. That's where we will find our taste buds, on the papilla. And it's not on the papilla, it's embedded in the papilla in the stratified squamous epithelium. So, can you see taste buds with the naked eye? No. You see papilla with the naked eye. You cannot see taste buds with the naked eye. There are 4 types of papilla on your tongue. The most numerous papilla on your tongue are called the filiform papilla. Then you have these guys called fungiform papilla, foliate papilla, and vallate papilla, or circumvallate papilla. Vallate's much easier to say, so they've actually shortened it down, but circumvallate was what they used to call them. Take a look at our tongue picture here. It's easy to figure out which is the tip of the tongue and the back of the tongue. Our circumvallate papilla are at the back of the tongue, and they form an inverted V back there. Our foliate are way back, and they're on the sides of our tongue. And then our fungiform are typically located at the front and sides of our tongue. The filiform are all over the tongue, and that's what gives your tongue its appearance, the velvety appearance. There are no taste buds in the filiform papilla. No taste buds in the filiform papilla. They are there to help us abrade our food. So if you take a look at them, they look a little spiky to me, and they help abrade the food. Why do you want to abrade or apply sandpaper to your food? You're trying to break it down, absolutely. So this is just a process of breaking it down because we're all born without teeth, correct? But we all have a tongue, and we all have these lovely filiform papilla that helps us abrade our food. So our notes here say the filiform papilla are short spiked, the anterior 2/3 of our tongue, and help with manipulation and abrade our food. And you want to break the food down too, because if you don't break the food down, how are you going to taste it? So let's now take a look at where the taste buds are located in the ones in the papilla that have them. In the fungiform, or the fungus-like papilla, they thought it looked like a fungus to them, taste buds are on the top. So those little bitty dots that you see coming off of what looks like CT, those are where the taste buds are. In the foliate and circumvallate, They're at the base of the papilla. So you have to go in the crevice in order to find those guys. Those are where the taste buds are located. And you guys are to look at the vallate or circumvallate papilla and make a histology drawing of that, of the typical taste bud. Let's take a look. We'll go to the notes in just a minute, because let's take a look at this picture here. What's this stuff here off to the side? Stratified squamous epithelium, correct? And then let's take a look here at the taste bud itself. We have a gustatory cell, and the gustatory cell is the neuron. We have supporting cells, which was just like what we had with olfaction. And then we have basal cells, which again is just like what we had with olfaction. And what do you suppose the basal cells do based upon what you learned with olfaction? Replace the gustatory cells. Because don't you sometimes burn your tongue? Yeah. So you do. You actually will burn and off your taste buds. But what's actually even more fascinating, your taste buds are replaced every 7 to 9 days. So taste buds are replaced, not taste buds, that's gustatory cells, excuse me. Gustatory cells, chemoreceptor cells, bulk of the taste bud replaced every 7 to 9 days. As you age, replace what, more or less often? Less often. Less frequently. Now, the gustatory cells have a hair that actually project out of the pore. Hair projects out of pore. Well, guess what's on the hair? Our receptors. That's where our receptors are located. So the hair, again, is not a real hair. It's a structure that looks like a hair, and that's where the receptors are located. Fungiform papilla, let's add to our notes now. We have buds, which when I say buds, taste buds, at top of the fungiform papilla. The papilla are in highest concentration at tip and sides of tongue. That doesn't mean they aren't over the surface of the tongue, but as far as density goes, tips and highest tongue. The foliate, we already know, are at the back lateral aspects of the tongue, and their buds at base of papilla. Circumvallate, also buds at base of papilla. These guys too are the largest. Oh, it just says in the notes, excuse me, we already have that down. And least numerous, and form an inverted V at base, which is the back of tongue. Let's go up real quick and take a look at some of this picture a little bit more. What are these structures off to the side? Right here. What are those guys supposed to be? Tonsils! Oh, guys, keep trying to make something complicated. And what's really interesting too, you don't realize it, but this is lymphoid tissue. Tonsils are lymphoid tissue, and we actually have a lot of lymphoid tissue at the very back or base of our tongue. You are never aware of that unless somebody rips your tongue out. I don't know too many people that happens to. Any idea what this structure is here? Just curious as to what your knowledge base is. This big guy right here in the back. That's the epiglottis. So that indeed is your epiglottis, something that whenever they intubate you, they are looking for that so that they can get into the right spot of your larynx. Quality discrimination is kind of what we were referring to earlier when we were talking about the wide variety of smells that we can appreciate, is also the same thing that we experience with taste. What's absolutely fascinating, we only have 5 taste possibilities. Yet when you experience food, it's not just 5 things that you actually have the experience of. So we only have 5 taste genes and qualities, and so it's obviously the combination of the activation as well as the amount of activation of these various taste sensations that we actually experience, the wide richness of our food. Now, if we are to appreciate sweet, the only thing you need to be is sugar or alcohol. There's more, but we'll just limit to sugars or alcohol. If you are a sour, all you need to be activated is by acids. Salty, those are inorganic salts, will activate you. If you are bitter, those are alkaloid chemicals. All you have to be is an alkaloid to activate. And then there is another taste sensation called umami. And umami is actually a taste receptor on cells that are not on the tongue, but they're actually on the soft palate and the back of your oral cavity and in your pharynx. And they respond to amino acids. Especially glutamate. And we even like the glutamate activation of umami receptors so much that we have created a salt shaker of MSG, monosodium glutamate. What cuisine uses a lot of MSG? Asian cuisine. So guess who actually discovered the umami receptors? It was Asians. So it gives you that meaty, or some people call the term, if you watch cooking shows, unctuous nature to your food. So yeah, like steakiness, MSG. Meaty appreciation. I call it bacon. Yeah, so that's activating our umami receptors. So remember, these are found in the soft palate and back of oral cavity. Anything that we need to take a look at with our cartoon of the taste bud or stuff along that line? Let me ask you something. So this is in the crevice, okay, right? So this is pointing out into the crevice, the hair with the receptor. And if you have a dry mouth, what happens to your ability to get to that little hair in that crevice? It's reduced. So if you have a dry mouth, you do not have as great of an appreciation of the food that you eat and things like that. So a lot of medications affect the oral cavity, the moisture in the oral cavity, and that's another reason why we like to have also lots of fluids with our meals. Yes, you actually can appreciate your lovely meal when you actually have a little extra moisture in the oral cavity to actually mix all those lovely chemicals around. And you're supposed to chew your food. Right? You're not supposed to gobble it, gulp it on down. You're supposed to chew it so you actually have an appreciation for it. Physiology of taste. Chemical must dissolve in saliva. So first off, you have to have enough saliva for it to dissolve in. And what do you suppose the foundation of saliva is? What chemical makes up most of saliva? Water. So it has to be water soluble. Must come in contact with a gustatory hair. That has everything to do with whether or not you have a dry mouth, and also whether or not you mix the food up in your mouth to get it down into the crevices by those papilla. Concentration of the chemical determines the intensity of the perception. So a little, not so intense, a lot, really intense for our perception, because we're only activating 5 different receptors, and the more receptors you activate, the more intense the perception. As far as sensitivity to those 5 basic taste sensations, bitterness is the highest, and actually we'll find a lot of those receptors at the very middle back of our tongue. That's why it's so much fun to try and swallow Tylenol. Anybody know what I'm talking about? Some of you swallow pills with no problem. There are those of us, and as you get older, it actually gets worse. As you try to swallow pills, it seems to be more of a problem. And so in order to get your pill down, you put it at the back of your tongue and you do your best to actually gush it back there with as much water as you can, or sometimes a straw works really well too. But if you're having a hard time and you can't, and no matter what you're doing, it's not going down and it's stuck on your tongue, you can taste the lovely acrid or alkaloid nature of Tylenol. And so that's why lovely children's Tylenol is flavored how? Fruity sweet, because the real chemical in that's pretty acrid, and so they are overwhelming them with sweet so that you can actually get that lovely stuff down them. Adaptation. Now, when you adapt to something, what does that mean to you with just that word, when you adapt to something? You get used to it, right? Like A&P. Yeah, you adapted. Yeah, you remember what it was like in the beginning. Yeah, it was pretty intense. Yeah, and you have adapted. So adaptation is a process that I depend upon for you in A&P, obviously. But this is also occurring with our chemical senses, taste as well as smell. Because you guys can know that you can walk into a room and somebody's farted. And you just go, oh, but you have to be there. But you know you will eventually get used to the smell and it will actually bother you less. This is true for taste as well as the sense of smell. So adaptation is a process. We get a rapid phase of adaptation for taste. In other words, let's have ice cream, vanilla ice cream. So we put vanilla ice cream in our mouth and the very first taste is the best, because every taste you take of that ice cream after that is never as good as that very first taste, because adaptation occurs. And then adaptation actually becomes complete in 1 to 5 minutes. So what that means is if you hold it in your mouth, after 5 minutes, you couldn't tell anybody what that taste was. Okay. And so this is a phenomenon that they actually use all the time in food and wine or food and beer pairings. Because what you can do is you can take a very acid wine and you can have spaghetti. What is spaghetti? Bunch of tomato sauce, right? Tomato sauce is acid. So you can take a very acid wine, but what you do is you taste it first and you go, oh, that's so acid, so strong, I don't like it. But then you'll eat the spaghetti, and then as you're eating the spaghetti, it's not that you're getting drunk off the wine, and so therefore no longer appreciate the wine. The acid in the spaghetti is causing the adaptation, and it's actually pulling that out of the flavor of the wine, and therefore you are now appreciating the other components of the wine's taste palette. And so they use this all the time in those episodes where people get together and do, as I said, beer and food pairing or wine and food pairing. So that's a very common phenomenon. We use adaptation all the time in those types of episodes or instances. Again, everything always starts as a receptor potential. In other words, chemical comes in contact with a receptor. That's a receptor potential. But until you get enough, you do not get a generator potential, because once you get a generator potential, you automatically have an action potential. You guys understand that, right? Receptor potentials always occurs first, and if it's enough, then you'll get a generator potential, which automatically means that we can actually figure it out or experience it. Now, taste is interesting. 5 basic receptor categories, okay? But yet it's so fun to eat food, yes? It's so fun to sit down to a meal and actually enjoy it if you're not just starving. If you're starving, all you want to do is just fill your face. But if you have time to sit down and enjoy a meal and sit and talk, you get to enjoy the food. But it's not taste that is just the only thing that you're activating, because 80% of our ability to taste is actually related to smell. What's at the back of our nasal cavity? An opening that goes where? Down the back of our throat. Well, what's at the back of our throat? An opening that can sometimes go up, especially when you're vomiting. Isn't that a wonderful experience when you do that? Okay. Goes out of your mouth and out of your nose. Correct. So I say these things mainly to actually wake you up and make sure that you're still with me. But also because anatomically we all know these things, but we forget these things when we're actually learning something. You already have a foundation of knowledge. I'm just asking you to add to that foundation of knowledge. So when you taste food, the reason that your mother wants you to chew it up and everything is that she actually wants you to appreciate your meal. Because the longer it's in your mouth and the more you chew it up, the more it will go backwards into your nasal cavity and actually activate and stimulate your olfactory receptors. And that's what gives you your appreciation of food. Because when you have a head cold, how good does your food taste? Pretty darn bland. And so you need that additional sensory input to actually have the appreciation for your food. So it's fascinating that what we equate to taste is actually a form of smell. So 80% of your taste is actually smell. Now, other receptors add to the richness of the experience. So what I'm telling you, in your oral cavity, there are more than taste buds, sensory receptors. There are simple sensory receptors. And can you give me some examples of some simple sensory receptors that would be activated by putting food in your mouth?
Mechanoreceptors.
What? Mechanoreceptors. Absolutely. Mechanoreceptors will respond to touch, pressure, stretch, right? Okay, mechanoreceptors. What's the one that causes pain? What do we call those receptors? Nociceptors, okay? And what about thermoreceptors? All right, so how many of you like soggy potato chips? Right off the bat. So it's the activation. Nobody raised their hand, just to let you guys know. So it's the activation not only of gustation and olfaction, but it's the activation of other receptors that provide you this wide richness of experience of enjoyment of your food. So other taste receptors, mechanoreceptors. Thermoreceptors. And nociceptors. Why do you want to have pain in your mouth? Because you like what kind of food? Hot, spicy food. You like Szechuan cooking. You like really spicy Mexican food with jalapenos and cayenne and all that kind of stuff. And some of you can't stand that, right? You just tell other people from now on at the dinner table, you say, I'm just really sensitive. Yes.
When you have a kiddo that has really problems with textures, what are you dealing with? Like somebody who can't handle it being very crunchy, or is it those simple receptors that they're dealing with, or is that are causing the problem?
Yeah. So if they don't like really crunchy food, they actually, those mechanoreceptors are more like nociceptors to them, and they have not yet developed the appreciation for that food. Now, what do kids do? What do infants do all the time? As they are developing and experiencing their world, what do they do with the things that they grab a hold of? They put it in their mouth. Your oral cavity has so many sensory receptors in it. It's really actually pretty incredible. And so they are using that experience of the item in their mouth, they're activating mechanoreceptors, nociceptors, thermoreceptors, as well as gustatory receptors in their oral cavity for that. So it's pretty darn fascinating. Now, eyes and vision. Fascinating. Eyes. 70% of all sensory receptors in the body are in our two eyeballs. That's a lot of sensory receptors. We already talked about simple sensory receptors, so we're actually talking about the construction of the eyeballs. Have 70% of all of our sensory receptors are located there. So that's why the loss of vision is so profound. Our experience of the outside world is so dependent upon vision. The only people that do not suffer the loss of vision are those that are born without vision, correct? So they learn about a world without their photoreceptors, and they have enhanced areas of their brain that when you're born blind, those other areas that you use to experience the world get larger. For us, our visual cortex is huge. Their visual cortex atrophies because it is not used, and the other areas that they are using to experience the world actually get larger and more sensitive. The eye is an interesting structure. It has accoutrements. It has a wardrobe, in other words. And so we call that wardrobe the accessory structures of the eye. And we have eyebrows. And sometimes you wonder why we have eyebrows. And the simplest explanation is it helps to prevent sweat from entering our eye. Some of us have very bushy eyebrows. You can tell some people their eyebrows are so good they have caterpillars on their forehead and nothing's going to get in their eye from their forehead. Yeah? Others, they pluck their eyebrows off and they pencil them in. Go figure. That's what it's all about. Eyelids, we have two for each eye, and the technical term for them is palpebrae. So if you go into any sort of field that involves the eyes or vision, you have to know this word palpebrae. Those are eyelids. And so with our palpebrae, or eyelids, our upper palpebrae, our superior and inferior palpebrae, we have off to the sides, medial and laterally, we have these angles called canthi. And so canthi is plural, canthus is singular. Another term that you must associate with that word is commissures. So lateral and medial angles of the eye. At the medial canthus, we find this thing called the lacrimal carbuncle. So that's a raised bump, and in that area, we will find sebaceous and sweat glands. So glands produce secretions, and so these are secretions that are actually meant to be a bit protective. What's sebaceous? What's the material from sebaceous gland? What's its oil? That's oily material, and you know when you put oil on stuff, less evaporation occurs, correct? And so you are able to retain moisture. Because the lacrimal carbuncle sometimes can get a little bit overactivated and overstimulated, it can produce what's called the sand or grit in your eye. When you wake up in the morning, you'll find a collection of that in those areas, and it's coming from that lacrimal carbuncle. We have eyelashes also associated with our eyelids, and they are innervated at the root, and this is because we need to actually have reflexive blinking, because we need to protect our eyes from foreign materials getting into the eyes. So if somebody is in a coma, and you know they're in a coma because when you touch their eyelashes, what will happen? Nothing. So they won't blink if they're in a deep coma. Now, if they're in a coma that's not so deep, they can sometimes actually have reflexive blinking because it is a protective mechanism. It is a reflex. Okay. Tarsal glands are also associated with our eyelids. They produce an oily secretion that helps to lubricate the surface of our eyes, and they are embedded in the area where we actually have our tarsal plates, and the ducts of the tarsal glands open at our eyelid edge. Now, tarsal plates are cartilaginous, or actually not cartilaginous, they are dense fibrous connective tissue structures that are in our eyelids, and they're the largest tarsal plates in the upper eyelid. There's a much smaller one in the lower eyelid. They help to give our eyelids shape and structure and form, and they're also something that little brothers like to freak their little sisters out with, because they have learned how to evert their eyelid and show off their little tarsal plates, to their sisters, and their sisters think it's gross, and their brothers think it's even cooler, whenever they can actually gross somebody out. I don't know about you. I actually did it to my brother because I was 10 years older, so it was more fun. But there are glands also associated with these tarsal plates in our upper and lower eyelid, and it's the oily secretions that they produce to actually help lubricate the surface of the eye. And why would we want to lubricate the surface of the eye? Because lubrication means that something's gliding along it, correct? What's gliding along the surface of the eye? Our eyelids, because we blink. And so we have the blinking that actually occurs, which is friction, right? So that helps to reduce the friction, and also the oily material also helps to keep moisture in with the eye itself. So keep that in mind. Conjunctiva. This is so much fun. This is what you guys usually can't seem to figure out on the cow eye and cow eye dissection. Every eye comes with conjunctiva. And conjunctiva is, for the most part, when you're alive, transparent, which means you can see through it. So when I say when you're alive, it's transparent, what's it like when you're dead? It's not transparent anymore. That's right, it becomes opaque. It's a mucous membrane, and it is very well supplied with blood vessels and glands, yet it's clear. So blood vessels and glands are all in the conjunctiva because it's a mucous membrane, but yet you can see through it, except when it's inflamed. And what do we call that situation when it's inflamed? Conjunctivitis. And that reason you see it is that the blood vessels dilate. So the blood vessels dilate and are therefore, when they're dilated, that means they're bigger in their appearance and therefore easily seen, and a lot more are dilated and irritated, and so then you actually can appreciate it. What's interesting about the conjunctiva is it is stitched to this area here, and it is stitched right around there.
Right around the ciliary body.
That's not the ciliary body. Where are we? We're on the outside of the eye. Where's the ciliary body? It's inside the eye. Right around, the iris. Is the iris on the outside of the eye? It's on the inside of the eye. So what is that I'm, cornea. Okay, so let's draw a side view of this. Here's an eye. There we go. Here's my eye. My pupil's in there, right? Okay. Here's my eyelashes so you can figure it out easier. Okay. So what I'm telling you, and here's the cornea, I'm telling you it is stitched to here and over here. Stitched to here and, oops, and to here. So that means if somebody took your eyelashes and went like all the way apart, you would be exposing all your conjunctiva, yes?
Yeah.
And you don't realize that it is actually lining the undersurface of your eyelids and the surface of your sclera, of the anterior portion of your globe or your eye. You don't realize it because you may not wear contacts. And it's because of this anatomical construction of this conjunctiva there that you can wear contacts. Because sometimes contacts displace themselves, yes? And it's not to the outside world. It's to underneath your eyelids. And so you have to raise your eyelids up, right? And you have to go looking around for those little guys. And the reason they aren't in your brain is because of the conjunctiva. You guys following me with this? So you don't realize it, but you do realize it, that the conjunctiva is keeping the outside world from your brain. Fascinating. So that mucous membrane is keeping the outside world away from your brain. So it's a lot tougher than you actually realize. Because the old note said delicate mucous membrane. I thought there's nothing delicate about the conjunctiva in my mind. And when you're in the lab and you're trying to actually manipulate the conjunctiva with the cow eye, you will find that there's nothing really delicate about the conjunctiva because it's going to be in your way and irritating. But it's a very excellent structure, very protective. So let's put in our note, lines, eyelids. Plus covers anterior surface of sclera. There's something that I didn't put in there. What does it not cover? The cornea. So if you had the conjunctiva covering your cornea and you got conjunctivitis, how well would you see? Not well at all. So it's actually a pretty smart idea to have the conjunctiva not covering that structure, which is vital for you to get actually light and an image into your eye. So, not the cornea. And this picture here, for when you refer back to your notes, goes to this stuff here. Okay. Lacrimal apparatus is the lacrimal gland plus ducts that drain into the nasal cavity. So what's really fascinating here, let me erase some of this stuff, is that the lacrimal gland is in the upper outer quadrant of the orbit of the orbital cavity of the eye. So your lacrimal glands are out here. Yet when you experience tears, where do you experience them? Medially, nasally. I'll use the term nasally. And so it's actually pretty cool because what that means is that the secretions that it makes, which are called tears, or we'll call them lacrimal secretion, but the tears is a very common term. That means that they run from a superior to inferior and medial aspect. So that's very good in effectively keeping the surface of the eye that's exposed to the world effectively washed and cleaned all the time. And then with your blinking, of course, that also helps to promote keeping that surface of your eye clear and free of all the nasty stuff, which will then get trapped in the fluids and then leave the surface of the eye into what area of your body? Your nose. So your tears are made all the time, and they increase their production when we're sad, or tears of joy, or hurt. So we increase their production when we undergo certain emotional events, and when we make the production higher than the drainage system will allow, we will then experience tears running down our face. Also, we'll experience a lot of sniffles, right? Because we're increasing tears that are actually running into the lacrimal sac, down the nasal lacrimal duct, into the nasal cavity. So tears are formed 24/7, and they're always dripping into your nose, but at a low rate, so you never actually really notice them. Now, tears get into the lacrimal sac first before they go into the nasal lacrimal duct, and they go into these two little holes called puncta. So lacrimal puncta, those are holes. And then the little ducts are called lacrimal canaliculi that actually take the tears to our lacrimal sac. Now, sometimes the lacrimal puncta can actually be blocked. And that's not an uncommon experience for some newborns. Yeah, and so their puncta have not completely opened, and if you don't completely open, where do your tears go? the tears go down your face. And so for a newborn that is having constant tearing, that tells you right off the bat that their lacrimal puncta have not opened up, and so the tearing occurs. Typically, their lacrimal puncta will actually open up within several months to, at the worst, 6 months. And if it doesn't open up, then you go to a doctor, and the doctor will open it up for you.
I have a newborn, and he doesn't, I mean, he'll cry, but there's no tears at all. Well, he's only a month old. So is that because the lacrimal gland is not?
He has very nice, generous lacrimal puncta, and or else he's just testing you a lot. There are different temperaments of children, everybody knows. Anybody that's had any. There's what you call the really good kid and the really, I wish you would go to sleep kid. You know, and never wants to go to sleep, wants to be continuously stimulated. Let's go to the next term of structures associated, the accoutrements of the eye, the extrinsic muscles of the eye. And there's an important term here. Extrinsic means external. And when you hear the term intrinsic, that means inside, internal. So we have extrinsic muscles of the eye are external. They're attached to the external aspect of the eye. And then we'll have some intrinsic muscles of the eye, which are our iris and ciliary body that we'll talk about in a little bit. We only have 6 muscles that control the position of our eyeball for each eye. And 4 of them are rectus. So we have 4 rectus and we have 2 oblique muscles. The rectus muscles are superior, inferior, medial, and lateral. So where you hear the word, that's where you find them in relationship to the eyeball itself. And when they contract, the superior rectus muscle will make the eye look up. The inferior rectus muscle will make the eye look down. The lateral rectus muscle will make the eye look off to the side. And the medial rectus muscle will make the eye look at your nose, right? When you look up, do you look up one eye at a time? No. So you have to understand the innervation to your rectus muscle is like a coordinated event. So when you look up, you activate both rectus muscles simultaneously. The same thing when you look down, correct? And the same thing when you look at your nose. But what about your lateral rectus muscles? Do you co-stimulate them? No. So with our lateral rectus muscles, what's really interesting, we stimulate one lateral rectus muscle and we inhibit the other and stimulate the medial rectus muscle in order to look to the left or in order to look to the right. So the rectus muscles do provide direction of the eyeball in the direction that's also named, but the only ones that are not co-stimulated at the same time whenever we look in a certain direction are our lateral rectus muscles, unless you're Marty Feldman. He's got those big bug eyes that look all over the place, or a chameleon, you know, so they can do that. So the rectus muscles move things all over the place. So move eyeball, and they're located in the area of the eyeball too, where they are stated. Move eyeball in direction of name. The oblique muscles are superiorly and inferiorly located, and here is our superior oblique, right here, this guy, and here is our inferior oblique, this guy. They perform complex movement. And they're a bit backwards. The superior oblique is down and out. The inferior oblique is up and out. So their names are backwards. So when you hear superior oblique, realize you're looking down and out, and inferior oblique up and out. One thing that I forgot to mention about the lacrimal apparatus, what does it produce? Tears. And what's the main component of tears? Water. Second main component, not surprising, is salt. We also have lysozymes, which are digestive enzymes, which help us destroy nasty things that get into our eye, and antibiotics. So we actually make our own antibiotics. Tears are to help cleanse the surface of the eye, as well as protect, which gives us moisture and lubrication.
So why do they cry into emotion?
We just have a parasympathetic activation of the lacrimal gland. So it's probably for socialization purposes. Because when somebody cries, socially, what do most people tend to do? Go. So, yeah. All right. That's where we'll stop today.